as well as DNA viruses. So in 1970, I'd spent about 10 years at that point as an investigator of the molecular biology of RNA viruses. And I realized that they fell into a pattern. And it was all built around the central dogma. So the central dogma of molecular biology, as we all know, is that DNA can duplicate itself. It can be the template for making RNA, and that the RNA can be the template for determining the structure of proteins. And then in 1970, in fact, I and Howard Temin discovered that one of those arrows can go back the other way. That is, RNA can be a template for making DNA. And so there is a movement of genetic information from RNA back to DNA and from DNA to RNA. Once we knew that, it was possible to classify all viruses within that structure. So they could have a single strand of DNA. Let's see, I can show that. A single strand of DNA as their genetic information, a single strand of RNA as their genetic information. That single strand can be the plus strand, the sense strand of RNA, or the complementary strand, which has no sense in the sense that it can't encode protein. But it can be copied into messenger RNA. And all of these kinds of nucleic acid can be copied into messenger. The one I didn't mention is double-stranded RNA, which is in some viruses. And so with all of these possible templates, the messenger RNA can be made actually in 7 different ways from viruses. When I first realized this, I defined 6 kinds of viruses. And later there was a 7th added. And we use this classification today. I'm amazed because in 1970, when I published this, I had no idea that it would stay on so effectively. And it shows that the virus world is much more complicated in the sense of genetic systems than the world of higher organisms, something that we might not have expected. Now, what are the sizes of RNA viruses? Well, here's a whole lot of them. You can't read their names, but it doesn't matter. Shown getting larger and larger and larger as we go from one class of viruses to another until they reach the end. And actually the end is right here, where the largest of viral genomes can be found. These viral genomes are segmented. Like flu, they have 8 different segments in influenza virus. So that's not a single strand of RNA, except for this one virus way out here. And that's the coronavirus, which is much larger than all of these others. So why is that true? It's true because the polymerases that make viruses are error prone, all of them. And the error frequency is about 1 in 10,000 nucleotides. So if a genome is 10,000 nucleotides long, or maybe up to 20,000 nucleotides, then the number of mutations is not so great per genome, 1, 2 mutations per genome. But if it gets any larger than that, the number of mutations gets to the point where it interferes with the effectiveness of the virus. And so a virus can't be any bigger than that without going through error correction. And the coronaviruses are the one kind of virus that has an error correction mechanism in it. Now, we in our own genomes have very effective error correction mechanisms. Our genomes get enormously larger than this because we have these error correction capabilities. All right, let me go now to the 2 scourges. The last 30 years have seen 2 viruses emerge that have really changed the world. HIV causing AIDS and SARS-CoV-2 causing the disease called COVID-19. Let's first focus on HIV. So it's what's called the lentivirus. Lenti means slow, and it slowly develops symptoms. In humans, that can be over a period of years, 10 years, but there are animal lentiviruses, and they're also very slow to develop their disease. And that's where lentiviruses got their name. They are retroviruses. They use reverse transcription because they send their genetic information from DNA to RNA and back to DNA and back to RNA. They grow only in one kind of blood cell. It's called the CD4 T cell or lymphocyte. And it's the only cell to which they can bind because the CD4 molecule on the surface of the cell is what they bind to. And this is a quick view of the life cycle of retroviruses, lentiviruses, and others. The particle itself, shown over here, has large numbers of proteins in it. In fact, most of the genetic information is for proteins that are in the virus particle. It also has 2 copies of an RNA molecule that encodes all of these proteins. And when it binds to a cell, it puts the RNA into the cell, and the RNA is then copied into DNA by the reverse transcriptase, which is in the particle. And that copy gets into the nucleus, and the DNA incorporates itself into one of the chromosomes of the cell. It turns from an extracellular agent into a set of genes in the cell. And then it's copied like any other genes in the cell into RNA. The RNA makes protein in the cytoplasm. The proteins that are made can come together to form new particles, and those bud off the surface of the cell. And so it's a complex life cycle, but it's well understood. And in particular, the reverse transcription process is very elegant, but I won't go into how that works. I'm just going to blow up this. Actually, I've been through all of this. Now, when the viral DNA integrates into the host DNA, there are 2 possible outcomes. Either the genes can function to make large amounts of protein, and the protein can then form large numbers of particles, but that probably will kill the cell because it's taking up all of the cell's macromolecular capability. Or the virus can only use a little bit of the capability of the cell, in which case it doesn't kill the cell. And now the virus can change the properties of the cell, and in particular can make it a cancer cell. And that's how cancer viruses in fact work. They go in and they continually produce the products of a gene in small amounts, but sufficient amounts to turn the cell into a cancer cell. It also means that if we use our skills as genetic engineers and put in genes that we want to put into a cell, we can carry out gene therapy. And today, there are large numbers of viruses that are being modified to carry therapeutic genes into cells. And gene therapy is a very active part of medicine. If the viruses get into germ cells, then they can be transferred to the next progeny animals or plants, but it's mainly animals. And if the virus is silenced so that it doesn't cause up anything like cancer, it can be passed silently from one generation to the next. In fact, this has happened extensively over evolutionary time, and 8% of the human genome is retroviruses of the sort we've been talking about, mostly silenced. In fact, mostly mutated to the point of being not very effective viruses anymore because they've been around so long. But